Okay, hello everyone. My name is Michael Downey, Library and Information Services Coordinator at the AWRI, and it's my pleasure to welcome you to today's webinar and also to welcome today's presenter, Dr. Paul Smith from the AWRI. Currently employed as Research Manager, Paul has been with the AWRI since 2003 and has a strong background in research into phenolic and tannin chemistry. Paul is currently involved in a project assessing the relationships between grape chemical composition and grade allocation grade, the results of which form the basis for the presentation today. Now, before I go to Paul to tell us more about today's topic, just quickly a couple of notes on what you as the audience should expect. The format is a short PowerPoint style presentation followed by a Q&A. The session is being recorded and will be available to view via the AWRI website. And finally, for those that are new to webinars, it's important to remember that webinars are interactive and that there are a number of ways to get involved. To ask a question, please type into the questions section of your control panel in the bottom right corner of your screen. Now, for those with access to a microphone, there is also the option of speaking directly with our presenter by clicking the raise hand button. This is located in the top left section of your control panel. If you'd prefer to ask a question via email, please do so at infoservices@awri.com.au. Please feel free to send through questions at any time, and once we get to the Q&A, I'll get as many questions through to Paul as possible. Finally, if you'd like to get involved through Twitter, please use the Twitter handle @_AWRI. Okay, that's enough for me. I'm now going to hand over to Paul to present his topic.
Okay, thanks Michael, and hello everyone. So I'm going to share with you today the results from some information on a project that we've been working on since 2013, and I'm going to cover just the data from 2013 and 2014 today, but at the moment the project has continued into 2015. I can tell you a bit more about that at the end. So we're looking at the assessment of relationships between grape chemical composition and the allocation grade that those grapes are assigned to. And whenever I use the term quality through the presentation, I'm going to basically be referring to the degree to which a wine achieves its intended style and the fruit that's used to make that. So what we focus on here is being aware that all the way along the value chain, from the vineyard soil all the way to the point of sale and beyond, there are a number of points where you can create style, and you can create it at certain points and you can preserve it and make sure it's not lost at other points. And so a key part of what we're doing in our research is trying to understand where that style is created and how you can create it with the least effort, really. So, as many of you know, there's a whole lot of general quality measures that are chemical based for production of wine. So they are things like free and total sulfur dioxide, the absence of turbidity and protein haze, balance of pH and TA, the amount of ethanol in the product and any residual sugar. But I'm not going to address those today because they are fairly central to the winemaking process. What I want to talk about today is some of the more advanced chemical measures that can be related to aroma and taste properties in wines, and where those taste and aroma properties come from in terms of the grape chemistry. I'm going to start by trying to orient you on where all these different chemicals come from and how they're created throughout the process. So broadly, we consider that the chemical measures of wine styles can be put into these four main classes. So the first is grape metabolites, or grape chemical compounds that are directly extracted from the fruit into the wine with no real change. The second is wine aromas, or flavours and tastes that are released or created from grape compounds, but without much help from yeast. So as soon as you start that winemaking process, they undergo some chemistry and transform and become the critical chemicals that you want in your wine. The third is wine aromas that are created primarily by the action of yeast on grape precursors. So obviously you kick off a ferment, and whether or not you use an inoculated yeast or just a wild yeast, that yeast can act on different precursors and turn them into new chemicals. The final one is the class of grape metabolites that are influenced by post-harvest processing and yeast effects. So here you've got the double whammy of things that you do in terms of crushing or how you treat it in the winemaking process, as well as specific yeast effects. So I'll walk you through some of the examples of these. So unfortunately there's a very small number of molecules which are directly extracted out of the fruit into the wine in an unchanged manner. So there are examples of these are rotundone. So this is the classic black pepper aroma in Shiraz that was discovered at AWRI 5 or 6 years ago. There are the class of compounds known as methoxypyrazines. These are the classic bell pepper if you're in America, capsicum if you're in Australia, green capsicum aromas, quite characteristic often of Cabernet. And then some from the class which we call C13 norisoprenoids. And a classic one here is what we call beta-ionone, and that's an aroma molecule which gives a characteristic violet aroma. So if they're in the grapes, they basically get extracted straight into the wine, and there you go, you've got your aroma or your taste product. So getting a little bit more complicated, wine aromas that are released or created from grape compounds, but not really from much involvement with yeast. So these include a class of molecules known as the terpenes, and some examples of these terpenes are compounds known as geraniol, which are in the skin, or linalool, which is throughout the berry, nearol, which is in the skin, alpha-terpineol, and these are all generally associated with kind of citrusy, fruity aromas. And there's another one known as wine lactone, and this is a classic example of the limey character in Rieslings, for example. Another one of these C13 norisoprenoids, similar to beta-ionone, but this one's called beta-damascenone, and it has an almost a cooked apple fruity aroma with it. And then C6 alcohols, which can give kind of cut grass type of aromas. So all of these are, have interactions once the grapes are crushed that liberate them through the process of either just the fruit being crushed or the wine being made, and that liberates these compounds from their precursors. Okay, so the third class of wine aromas created by the action of yeast on grape precursors. So an example here are fatty acids, and these often can give cheesy, sweaty kind of aromas, generally not that pleasant. Esters, so esters quite often are formed from precursor amino acids through the action of yeast, and esters are characteristically fruity of different types. There are some higher level alcohols which can give sort of fusel character or some higher level alcohol character. 4-ethylphenol is a classic example, of course, of a spoilage molecule. So this is formed by the action of Brettanomyces on phenolic precursors and gives that classic band-aid, horsey, barnyard kind of character. And volatile acid, so that's obviously another negative one, which is caused by the action of spoilage bacteria. Okay, and then finally the quite complicated class are the grape metabolites influenced by post-harvest processing as well as yeast effects. So examples here include things like what we call the varietal thiols. So these are the molecules characteristically responsible for Sauvignon Blanc aroma, like the passion fruit, box hedge kind of character, but we're also finding that they play important roles in varieties like Chardonnay and possibly even some red varieties. And these can have precursors that are created through the crushing process, depending on how long you hold the juice once it's crushed, and then also depending on the yeast you select, the release of these precursors can be highly variable. And so, yeah, yeast selection we've shown is very important too for creating this varietal style character. Tannins, another very complicated class. So tannins are in the skin and the seeds, obviously, and during the extraction process, depending on how much skin contact or seed contact you have, temperature, ethanol concentration, so on, that'll depend on how much tannin you get out and how much tannin comes out of the skin or the seed. And then once that's out, we've shown too that your yeast selection can have a profound impact on the concentration of tannin that you end up with, as well as the actual chemical structure of that tannin, which has knock-on effects to its mouthfeel. Colour is a classic one, and the formation of pigmented polymers and non-bleachable pigment, strong long-term colour, is influenced by yeast effects and also by how much processing and so on you do and the way you process the fruit. And then finally, a class that we're really starting to dig into now is the polysaccharides. So these can be small, medium or large, and they are polymeric chains of sugars, which are generally associated with weight and viscosity. And they can come from either yeast or they can come directly from the fruit. And for example, the type of clarification processes you use or the yeast selection that you make have a big impact on these classes. So hopefully that orients you about the types of chemicals that are in wine and the kinds of points that they can be influenced at. There's one other class that I won't touch on much, which is the environmental class. So these are compounds which come in directly from the environment. Classic example is a eucalyptol character from 1,8 cineol, and work here from the flavour team has shown that can often come in through MOG, so leaves and bits of bark and things that are caught up in the fruit at harvest point. Smoke taint's another example, with smoke drifting across a vineyard and being absorbed into the vine. Other general taints, and now there's some research going on about what other regional plants might be contributing to aroma and taste profiles in wines. Okay, so what's the context to all this work? Well, as I said, many individual compounds are reported to have relationships with the grades of grapes, with wine grade, and also relationships with sensory. I've given you a bit of an overview there of some of the ones we know. If you'd like to read about that in any more details, I wrote a proceedings article for the Australian Society of Viticulture and Enology in 2012, and I'm sure you can ask Michael for that from the library. So, as most of you know, colour has been historically used in Australia for grapes, and it's still used to varying levels by varying companies. But it is generally a strong predictor of grades, particularly for warmer region fruit. And we've established the AWRI over 10 to 20 years, even 30 years back when Summers was around, that colour does have strong relationships with wine grade. And then more recently, work out of my team has shown that tannin concentrations can have strong relationships with wine grade as well. And there are companies around the world using varying degrees of those chemical measures I mentioned earlier for various applications. So Gallo are reasonably open now about the fact that they stream their fruit using an index of multiple compounds. So it's not just one compound, they look at the relative proportion of many of them. In Germany, there are cooperatives looking for released aroma potential from a measure called the glycosol glucose assay that was developed here at AWRI. There's companies in Portugal using tannin measurement and colour measurement for fruit streaming and for winemaking decisions, and then various players around the US, New Zealand, Italy, Portugal and South Africa using tannin measurement and colour for winemaking decisions. So what we're talking about today is not an abstract thing, and it's very much being adopted around the world. So our project question from an Australian context, our project questions were the following. regardless of the grade that the producer gives to the fruit, can chemical measurements be used to essentially cluster fruit to identify similar patches or highlight differences between them? So is there a chemical fingerprint that might give us a clue about how to stream fruit that's similar to each other? That assumes no knowledge of the grade, and yeah, the application is that it could be used to stream fruit. Now, I don't have much time to talk about that today, but I'd welcome any emails or questions at the end if you want to know more about that, and I'll flag some articles too. So more of the focus of today will be, can existing grading allocations be predicted using some of the previously identified chemical measurements? So that assumes that the current grading system is accurate and it seeks to try and bring some objectivity to the process of assigning grade to grapes with the support of chemistry. So, okay, on to the details. What chemical analysis did we do? So berry basics, things like average berry weight, the pH, the TAs, Brix, the percentage of moisture, malic acid, alpha amino nitrogen, ammonia and YAN, 3 different ways of measuring the available nitrogen for yeast to feast on. We looked at some possible negative markers too, so lacase activity is an enzyme that comes from some of the spoilage fungi like botrytis and so on. We did measure those, but we actually didn't find very much in the fruit that we got from these. Chloride is a possible marker of salt pickup and has been shown to have negative impacts on sensory. Finally, we did a whole range of spectral measures. So things with simple UV-Vis spectral instruments that many wineries have got access to. So measures like A280, which give you the total phenolics. Measures like 520, which give you the colour. Measures like 420, which give you an idea of, for example, brownness. And we also measured tannin using a method that we use called the MCP tannin assay. Then we measured some aroma compounds, and these classically require some more sophisticated equipment, but there are still well-established methods for measuring these. So compounds like the grassy C6 compounds, methoxypyrazines, which give that green capsicum, and then this one called the glycosol glucose assay out of grape homogenate, where we just blend up the grapes. So what this does is basically measure the released glucose from possible aroma precursors. So it's a kind of total measure of potential aroma. And then we measured one of these fruity aroma markers, free beta-damascenone. Okay, so I'm going to start by summarising what we found in 2013. This was a pretty quick kick-off to the project. So we did just Cabernet in 2013 as we were ramping up. And what this graphic shows here are some relative positive or negative loadings on specific chemicals. So along the bottom axis, you can see a range of different chemistries that we measured. And then the axis that goes through 0, above that is positive, below that is negative. So what I've just brought up now was discovered on using a statistical approach called PLS regression modelling. And so it was a way of trying to predict the grade using the chemical data, and it's particularly well suited because it gives us some clues about what chemical compounds are actually allowing the discrimination between the grades. And so the ones I've just highlighted here in green, in 2013, basically there were higher amounts in better grades of fruit in that year. So they were things like a couple of the amino acid nitrogen measures, so alpha amino acids and YAN were in higher amounts in better grades. And then some of the classical UV-Vis spectral measurements, total phenolics A420 and A520, which is responsible for colour. And then this beta-damascenone fruity molecule. Then we also found that there were some, what we call some negative markers. So there were lower amounts of these in better grades, or conversely higher amounts in poorer grades. the TA came up there, and then three random amino acids, cysteine, glutamate, and glutathione, and we can't directly assign them to any particular sensory attributes directly, but we know they do have some roles in the winemaking process. So using all those grades and all that chemistry data, we were able to build a model where the PLS regression was able to predict an R-squared of around 0.49. So that means that about 50% of the variability in the data could be described by this model. So that's sort of a moderate prediction. It's not great, it's not terrible. So the take-home message out of 2013 was positive. We had a moderate ability to predict grade using chemical data and this statistical approach of PLS regression. It suggested some lead targets in terms of chemistry to keep an eye on in the 2014 sample set. So they were YAN, total phenolics, absorbance at 420, absorbance at 520, so the red colour, and free beta-damascenone as being positively associated, i.e. higher amounts in higher value grades. And flagged some possible negative markers, so TA, cysteine, glutamate, and glutathione. So then we pushed on into 2014, and a key point about 2014 is that we were looking at now Cabernet Sauvignon, Shiraz, and Chardonnay. And this was, I should say, this was all fruit that was provided to us by Accolade, our primary collaborator, and Fable Vineyards. And so the fruit came from a wide range of regions. A lot of it was from the Riverland, but we also had representatives across all the varieties, variously from Swan Valley, McLaren Vale, Langhorne Creek, Clare Valley, Padthaway, Coonawarra, and Rattonbully. For Chardonnay, we additionally got fruit from Adelaide Hills, Great Southern, Margaret River, Pemberton, and Tasmania. But not all regions had all three varieties analysed. So we ended up with around 60 parcels of fruit, at least, from each variety, spread across those regions. So again, what we've got here are an example of the PLS predictions with the chemical loadings for Cabernet in 2014. So what we found was a wider range of chemistry marked as being positive in 2014. So there were compounds, a range of different compounds that were amino acids, which can play a role in yeast nutrition, but can also potentially be precursors for aroma molecules such as the esters, and can also sometimes be stress markers. And then a whole range of the spectral measurements as well, so 280, 320, 370, 420 and 520. They are total phenolics, hydroxycinnamic acids which come from the pulp, flavonols, which are generally sunscreen exposure markers, 420 tends towards brownness and 520 is redness. And then as negative markers, we found YAN in 2014, and I should note that that's flipped now from 2013, because in 2013 it was actually a positive marker, and that might be because in 2013 it was actually low, and in 2014 it was quite high. So that may have just changed that a bit. So there are a couple of other nitrogen type measurements, and then down the end here these two Z3 hexenol and E2 hexenol, and so these are two of those grassy C6 compounds which were loaded negatively. So by bundling all that in and doing some predictive regression, we were able to predict grade by an R-squared of 0.65, so 65% of the variability predicted. So they were generally amino acids, UV-Vis, tannin came up positively, and also glycosol glucose, which was that aroma precursors measure. And there was some influence of the C6 compounds negatively. Yeah, so there were some other molecules that were flagged as positive, tannin, glycosol glucose, and Brix. Now another way of trying to get strong predictive capabilities or predictive models is to use what's called discriminant analysis, and this uses the principal components from another statistical method called principal component analysis. But it takes all that data and it builds a model that allows you to predict the grade of the fruit based on that chemistry. The challenge with this is that it doesn't allow you to really drill down and find out what chemical markers are allowing that prediction, which is why we use PLS regression on the previous slide to look at under the hood and at the chemistry. But anyway, what this shows is that we had very strong ability to predict grade using discriminant analysis, and that's probably because discriminant analysis is very good for categorical prediction, and really the grading process is a categorical process, not a linear one. It's just a A, B, C, D, not a 1, 1.1, 1.2, 1.3 type of thing. So what you can see is down the diagonal there are all the ones which were predicted correctly, and anything that's flagged in red was one that was incorrectly predicted. So we get about 90% accurate prediction for the Cabernet data in 2014, which is very, very good. So what about Shiraz in 2014? It's the first time we'd looked at a wide range of Shiraz, and again it came up with quite a large number of positively loaded chemistries. So some of those nitrogen markers, YAN and alpha amino nitrogen, were positive. A few random amino acids, which we're not sure why they necessarily would have popped up or what their functional role is, but then a number of the classic UV-Vis spectral measures, absorbance at 280, 320, 370, 420 and 520. And then in Shiraz, positively loaded were two of those C6 compounds, so hexanol and E2-hexenol, these grassy green aromas. Tannin was positively loaded, and for some reason chloride was too. The only negative load that we got for 2014 was aspartic acid. I can't account for why aspartic acid might be a negative marker, unless it's potentially something to do with the stress indicator. And again, we got about an R-squared of 0.65 for this, reasonably good using PLS regression for predicting grade. And the variables that were discovered were similar to Cabernet, except for Cabernet C6s were loaded negatively, and here in Shiraz C6s were loaded positively. Again, if we look at discriminant analysis for the Shiraz data, we get around 85% accurate prediction, and so that's very good. And here's the confusion matrix again, and so those things along the diagonal show the prediction that is correct, and anything flagged in red are the incorrect predictions. So it's a very strong model for predicting grade using the chemistry. So finally, we're looking here at the Chardonnay from 2014. This is the first white variety that we had a really good close look at. And yeah, a little bit more complicated here, but pH was loaded negatively. So higher pH was lower graded. TA and ammonia and malic acid were positively loaded, as were now some of for the first time, the varietal thiol precursors. So this cis-3MH and cis-gly-3MH are precursors which, once they're acted on by the yeast, are released to give some of that classic passion fruit tropical character. And so it was interesting to see them being positively loaded. Negatively loaded was one of these green C6s, but then E2-hexenal. But then positively loaded was another two of these C6 precursors, so hexanol and Z3-hexenol. So there's a little bit of a mixed bag there. We know that sometimes these C6s can actually be precursors for the varietal thiols, but then they can also have that grassy green aroma, which if there's too much of, can be negative. So that may be why we see a mixed loading there. And then the glycosol glucose assay as a predictor for aroma precursors came up positively, as did chloride, again for some reason, I'm not sure. And then finally, negatively loaded was the absorbance at 370 nanometers. Now that's quite interesting for Chardonnay because 370 nanometers is flavonols, which are generally the sunscreens for the grape, and so they're often markers of exposure and potentially overexposure. So the predictive ability for the PLS regression here was better than for the reds. It was around 0.78. So yeah, quite a good model. And C6s, as I mentioned, were both positively and negatively associated with grade. And there were some interesting compounds, including proline, thiol precursors, glycoside glucose as an aroma precursor measure, and flavonols being that exposure marker. So finally, we looked at discriminant analysis using chemistry on the Chardonnay data, and again, very, very strong predictive model here, 93% accurate prediction for classifying the grade of Chardonnay fruit from 2014. So that brings me pretty close to the end, and I guess I just want to leave you with a couple of take-home messages, which are that overall, these results demonstrate that there are definitely a subset of chemical measures on grape that can be performed on grape homogenates or juice using a modelling method called PLS regression that gives promising technique for assessing and predicting grade, but also using discriminant analysis allows even better models to be developed. But from the discriminant analysis models, it's more difficult to understand what chemistry is driving that grade prediction. One thing that I haven't had time to talk about today is that we've also done a lot of work with Bob Dambergs and our other collaborators on using spectroscopy to predict classification, and that is actually very, very effective also. And the beauty of using spectroscopy for predicting grade and things like this is that it's relatively low cost and quite rapid compared with some of these other chemistry methods. And you can actually use spectroscopy to build calibrations to predict many of those chemistries as well. But yeah, I haven't had time to dive into that today. So for those of you who are interested in trying to dig deeper into this, I've written some papers recently summarising the work. So there's an article on the 2013-2014 Cabernet work in the AWRI Technical Review, the August 2015 edition. There should be a Grape Grower and Winemaker article coming out in September, which covers just the 2014 data, so Shiraz, Chardonnay and Cabernet. And that's in the September issue. And then later on in the year, in November, December, there'll be an AWRI report in the Australian New Zealand Wine Industry Journal. So I guess I'll finish by emphasising that all those chemicals that we measured there, some of them are reasonably straightforward to measure. Some of them do require, a higher level of sophistication, but some of those more sophisticated ones are also available commercially, so you can get someone else to measure them for you. And the key point is that at the moment we're looking right at the front point here of that value chain, the grapes, and it's just important to remember that all the way along that value chain, what you do to the wine can affect that different chemistry. So while we've been looking at grapes, now in 2015 we're actually making wines out of those grapes and seeing whether the chemistry that predicts grade is actually predictive of final wine quality and style, but particularly style. So we're looking at the compounds that are in the fruit and whether or not, if they're high, they do translate to more varietal thiol aroma, or more peppery Shiraz, or those kinds of relationships. So yeah, that just leaves me to thank the wide number of staff which have been involved in this project, because it's drawn on a lot of skills across the whole of the Institute. Lee Francis is a co-chief investigator with me. He's given a lot of the leadership to the aroma side of the research. Karen Binden helps out all the time with tannin, and then a number of people have been critical to providing the measurements. So Alex Shulkin's done a lot of the project management, Jackie McRae's helped, Bob Dambergs has been critical to helping us with the chemometrics and training us. And then Sheridan, Mark, Dimmy, Stella and Vise have all helped on the bench. But also finally a big thank you to Accolade Wines, particularly Warren Birchmore and Alex Sass, who have supported us with this project and always been incredibly encouraging and helpful. So thank you, and happy to take questions. Thanks also to now Wine Australia for their funding.
Thanks for that, Paul. We'll move from here straight into a Q&A. So start sending your questions in now. Just a reminder, if you're typing your questions in, please do so at the bottom right section of your control panel. If you've got a microphone and you want to chat directly with Paul, click the raise hand button and I'll unmute your microphone.
Okay, so thanks, me again. I've got a question here from Warren. Are there any results from 2015? So the answer is no, but what I can tell you is that we've basically got samples, a smaller number of samples from Cabernet, Shiraz and Chardonnay in 2015, and what we've asked for the support from Accolade for is to basically tell us what they think are 4 wine style categories for each of Shiraz and Chardonnay. Sorry, we haven't done Cabernet. Shiraz and Chardonnay. And so we've got 4 representatives of grapes that we think will stream into one style, and we're measuring the chemistry of that fruit, and then we will look at the chemistry of the wine and try and marry up the fruit chemistry with the wine chemistry, and make sure that the sort of chemistry that we're thinking predicts grade in fruit actually translates through to, you know, predicting a wine style or an appropriate wine grade. So we've just had the wines bottled, so the wines have been made. We've got the chemistry data from the fruit, but until we have the wine chemistry to marry them up with, we'll just have to wait and find out, you know, how predictive those chemistries from the fruit are of the actual wine style.
Thanks for your question there, Warren. Hopefully that's answered it. Don't have any further questions at this stage. We'll give it another minute or so and see if any final questions come through. In the meantime, Paul, did you want to, oh no, we've just had one come through, so I'll send it through to you now, Paul.
Okay, hi Kirsten. So Kirsten Creasy from New Zealand's asked, is it likely from this research that we'll see analysis of amino acids being part of a regular juice panel? Good question. I don't know. Certainly amino acids have been, there's a fair bit of research in the scientific literature about the role of amino acids as predictors of or playing a role in leading to ester formation. And obviously they are caught up in their totality, in yeast assimilable nitrogen and some of those kind of measures. I'm not an expert on the aroma side of things, so Lee would probably be better positioned to answer this, but I do know that there is a reasonable body of evidence that key amino acids translate into key esters. So yeah, certainly we're thinking that amino acids are something that we would measure in a standard way for our research. The question is also whether or not some of those amino acids may actually be markers of stress, for example. And then also some of those amino acids, for example glutathione, are actually precursors that get built into other compounds. So some of the mercaptohexanol, the varietal thiol precursors, can be influenced by the glutathione concentration. Also, there's another molecule known as Grape reaction product, which occurs when some of the phenolics from the pulp are oxidized. They then react with glutathione and form Grape reaction product, and that actually helps in some ways to protect the wine, particularly white wines, from browning. But we're also finding now that it may have some actual roles in itself around hotness and oiliness. So I think that there probably will be a time when amino acids are a regular, part of analysis. I'm not sure if we're there yet.
Okay, thanks for your question there, Kristen. That looks like it may be it with regard to the Q&A. Before we start to wrap up, Paul, did you have anything you'd like to sort of re-emphasise before we start to finish?
No, I guess my main point is start to have a think about using these metrics. Find some simple ones. If you've only got not a lot of resources available to you, find some simple ones, get interested in them and try and understand how they impact the fruit that you get from different parts of the world and different parcels of the regions that you work with. I guess I'd be curious to know whether anyone already is using some of the things I mentioned to try and understand their fruit. So things like tannin and colour, whether or not people are using them actively. If anyone wants to share that, that'd be great. But yeah, I'm happy to take emails from people down the track as the work evolves. And I think it's just important to note that if I'm brutal about it, Australia is falling behind because the rest of the world is using these chemistry metrics. Some of them are only just sort of realising they can use them, and the way they use them is varying depending on the organisation. But yeah, they are real opportunities to create value in the wine production system and make sure that you don't lose high quality fruit in with low quality fruit, for example, or that you really stream, for example, Sauvignon Blanc fruit with lots of varietal thiol precursors. You stream them all together and make sure that you make the most of them, and you don't accidentally blend them out with something else which is really low. And very close to my heart, of course, is tannin, and I think that pretty well everyone should be measuring tannin on fruit and on wine now, and really understanding how that drives their colour stability and mouthfeel and things. So yeah, like I said, feel free to email me, and thanks for your interest today. Okay, so I've got a question here from Marcel. Which markers could be measured 2 to 3 weeks before the predicted harvest date and still be meaningful? Yep, so that's a really good question because most of these things that we measured there were in the harvested fruit. So you can get a pretty good indication still of tannin. Tannin will change. The extractability, the total concentration of tannin won't change very much in fruit as it progresses, but the ease of extraction can change as fruit matures, and the general rule there is that skin tannin becomes more extractable and seed tannin becomes less extractable as maturity progresses. But a couple of weeks out, you won't be too far off the mark, but you'll have an indicator in terms of benchmarking this year's fruit against previous year's fruit. You might not have the final answer. So a number of places I know that use these kind of measurements do so in the week, basically the 1 week prior to harvest, and they monitor on a weekly basis leading up to harvest. And when they reach the metrics they want, then they make their harvest decision. Colour will be not too bad as a predictor. What else? Some of the aroma compound precursors do turn up pretty late in maturity. So some of those will be things that you need to do as close to your harvest point as you think you can get away with. Malic obviously changes with maturity, and that popped up a few times. Yan is something that you should be monitoring pretty well anyway. Berry weight will be changing a bit. So the closer that you can get to the potential harvest, the better, and really monitoring it potentially on a weekly basis is the best case scenario. But obviously there are resource implications for that. Okay, I've got another question come in from Kirsten in New Zealand. So winemakers in New Zealand are using YAN measures 2 weeks out to help with intake and processing decisions. That's great, thanks. Yeah, so that's sort of reassuring. And we certainly know that YAN is one of the critical things associated with quality, and that it tends to be one which is a bit more of a sweet spot measure. So you don't want too much, but you don't want too little. So the classical sort of 200 to 300 range, I think, is the happy point in general. And if you overshoot the mark, then you can have some aroma impacts, and if you undercut the mark, then you can have stressful yeast and unhappy ferments. But yeah, YAN's a really good one to monitor.
Okay, thanks for going through those final couple of questions there, Paul. That looks like it's it for today, so I'll start to finish up. Firstly, a big thank you to Paul for coming in and presenting today, and also a thank you to everyone who's joined the session and participated. For attendees, you'll receive a follow-up email with a link to this recording and a link to a survey. Now, should you need to follow up or have any further questions regarding this session, please contact Paul or the AWRI directly, and we'll forward inquiries through to Paul if need be. Paul also flagged a couple of research articles. If you'd like to get hold of them, please contact the library at infoservices@awri.com.au. Next week's webinar is coming from Tony Profit from AHA Viticulture and Precision Viticulture Australia. And the topic is using unmanned aerial vehicles in the vineyard. Now, if you haven't already registered for this webinar and would like to do so, please visit the AWRI website. Once again, thank you to everyone for joining, and please check out the AWRI's webinar program to register for future sessions.